Hello everyone, I hope you're doing fine in this pandemic. In this video, we will discuss about the different vaccines that are being given to people in India, what are the mode of action and what is the difference between all of them. So in this video, we will be discussing about the three videos, that is Oxford AstraZeneca based Covishield, Bharat Biotech's and ICMR backed Covaxin, and the Russian vaccine that is Sputnik-V. First of all, we should know that how the virus enters our body. The virus surface protein is 3CL probe which interacts with the ACE2, which is the most likely site for the virus inoculation in our cells. So basically what happens is this spike protein comes in contact with this and this virus enters our body. Then the virus hijacks our body. Basically what happens is the outer shell of the virus is basically protein and inside it there is an RNA sequence. When it enters our body, it injects its RNA sequence in our cells, and that RNA sequence is then transferred to the ribosomes where it does the protein synthesis. The protein synthesis is not for us, the protein synthesis it's for itself. Basically it manufactures its own surface proteins, its spike proteins in our ribosomes. It hijacks our cells and in that way it does its replication. So one should know that the virus is a very simple molecule. It just has a coat protein and a nucleic acid inside. So being that simple, the multiplication is very fast because it don't need many constituents to make its copy. So basically the most important thing that you should know is the protein that interacts. This 3CL Pro is a protease. Protease what does? They does the hydrolysis of proteins. That's why it is able to enter in our cells. So not just these three vaccines, the basic principle for any vaccine is that we give a weakened virus to that person. Even the polio vaccine, this thing was being done. So in SARS-CoV-2, what we do is we have a coronavirus. This is the general principle of vaccination. We will be discussing about the three vaccines in the later slides. So basically what is this? We have a coronavirus. This is the nucleotide and these are the spike proteins. We can give three types of treatment, that is chemical treatment, UV treatment or heat treatment. These treatments are given to destroy the nucleic acid inside it such that it won't be able to replicate itself. So basically what we use is beta propiolactone. The structure is shown this. This is a sweet colorless liquid. What it does it's bind to the nucleic acid and the nucleic acid sequence is destroyed. The spike proteins are present as such but inside of the nucleus is destroyed. So now when we inject this virus inside a person, what would happen is it won't be able to make it copies because the nucleic acid sequence is destroyed. But when it enters in our body, this is the human cell, when it enters in our body, it will interact with the these receptors on our cells like ACE2 or TMPRSS2, and our body will recognize it. Our body will recognize any foreign body that enters itself and would produce antibodies against it. So these are the antibodies formed for SARS-CoV-2. So these antibodies are specific for the coronavirus spike proteins. So basically what would happen is when original virus would come to infect us, these antibodies would go and bind to this spike proteins. So these spike proteins would be blocked and the virus would not be able to interact with the ACE2 or TMPRSS2. In that way, we would stop the virus from spreading in our body. So if we see that if we have some other medicine apart from vaccination of these three vaccines, if we have some other medicine to block these two sites, the spread of the coronavirus can further be stopped. This is a much better representation to show what's basically happening is these are the two receptors on our body, on our cells, and here comes the SARS-CoV-2. The SARS-CoV-2 binds with these receptors and the receptor is lost and the SARS-CoV-2 enters in our body. So by some means, if we block this receptor, the COVID won't be able to interact with it and it won't enter in our body. So we can either block this or this. So three things can be blocked to make it functional, to get any therapeutic for it. So being a chemist, I was more interested in the chemical structures. These are the structures of some molecules that could be used for this purpose. And these are the 3CL Pro inhibitors, and this is for inhibiting the ACE2 proteins that is present on our cells. Now come to the vaccine. So the first vaccine is Covaxin. The code name for Covaxin is BBV152. It is an inactivated form of virus. As I showed you earlier, that we have taken the virus, we have inactivated it by some treatment such that it won't be able to multiply itself in our body. So that way Covaxin works. It was developed by biotech, and the efficacy is important. It is around 80% effective, and it is similar to the polio vaccine. Polio vaccine was also made in this way, and you should know that this is the molecule that is used to deactivate the RNA sequence. It basically binds to the genes of RNA. So this is again that PPT that I showed you earlier. Now comes the Covishield. The mechanism of action of Covishield is slightly different from that of Covaxin. So basically what Covishield does is, we take an adenovirus. Adenovirus is a virus that causes flu-like symptoms like headache, running nose, and throat sore. So it is not deadly, but it causes flu-like symptoms. So we take a adenovirus. Adenovirus is an RNA-based sequence, means it has a double helical structure, whereas coronavirus has a single helical structure. So we take an adenovirus. With the recombinant technology, what we do is we modify its sequence such that it produces the spike proteins that are similar to COVID. What we did is, we took the adenovirus, we modified its genetic material in such a way that the surface protein now produced are similar to that of coronavirus. Now we inject the person with this adenovirus. Now the body recognizes the surface proteins, creates antibodies against it. So when in the later stages we are infected with the coronavirus, those antibodies will attack this coronavirus. Why we are using adenovirus? Because it is already weakened and we know that it won't cause us much side effects. It is having flu-like symptoms and can be treated with paracetamol or some mild medication. So we can see this is a chimpanzee adenovirus. Efficacy is around 80%. Okay. It inhibits the entry of the coronavirus inside the cell through ACE2 inhibition. We have already discussed it. Now comes the Sputnik-V. Sputnik-V has the highest efficacy so far, that is 91%, and there's a reason behind it. So let's discuss it. That Sputnik-V again uses an adenovirus. We can see that there is a double helical structure, whereas corona has a RNA sequence that is single helical. So Sputnik-V was developed by a Russian vaccine. This was probably the first vaccine that came to the market. It got approved in August 2020. It wasn't approved, but however the Russian government gave permission for general public to get it. So it is again an adenovirus vaccine, but is different from Covishield. It is different from Covishield in what aspect is that in Covishield, the two doses are of the same injection. We get the same coronavirus 2 times. But in case of Sputnik-V, they gave two different variants of the adenovirus, that is 26S and 5S. Both are adenovirus, but there is a slight difference between the two. Actually, they did a study. In the study, they gave some patients same dose of 26S. To some patients, they gave the same dose of 5S. And in some cases they gave 5S and 26S. This showed the best result when they gave these mixed doses. That's why they took it to the clinical trials. So basically everything is same, but they just give two different doses of the two different variants of the adenovirus in 2 different doses. It is also in news that Sputnik-V and Covishield are planning in such a way that it is not necessary to have Sputnik-V. If a person has Covishield as a first dose, it may also have Sputnik-V as the second dose. I read it somewhere that it is under consideration, so probably we get to hear more about it. So this is it for this video. I would also like to add in what way Remdesivir is helping people. So if you see here, that basically what happens is that the coronavirus RNA sequence enters in our body, uses our rhizomes, makes proteins, that is the corrode proteins, the spike proteins itself, for itself, and make its copy. So what basically Remdesivir does it, Remdesivir targets the protein, a gene called RDRP, RNA dependent RNA polymerase. In what way happen? The DNA, the RNA replication is stopped. Once the Remdesivir attacks the RDRP gene, the DNA replication is stopped, and it also targets the viral exo enzyme. So it delays the replication. So basically Remdesivir is acting in that way only. That's a different topic for a certain video, and you should also know that all the data I've taken is from other sources, so I take no liability for it. Thank you.